So if you go to allencell.org, you can see that if you click on the Allen cell collection, you'll see our mega menu, which you can select different things. We're going to first start with our cell catalog. If I click on that, you can see there are always updates, the latest updates of what new lines are available. And then we can scroll down and you can see the different list of the proteins that are tagged. You can find information of which, where the tag is located, so on the C or in terminus. You can also filter by what structure you're interested in. So let's say that we're interested in a structure. I can just pick arbitrarily like microtubules because you've seen the images of that. And you can see there's two, it narrows down to two. One, the difference is that one contains a GFP tag and one contains a red fluorescent protein tag or RFP tag. And so we have tagged some of our lines with RFP, but most of them contain a GFP tag. So if I click on this link, it's going to bring up a page. It'll have a Z-disk, a Z-stack, sorry, of that cell line, and then I'll have information on the left that contains everything from a data summary of our QC analysis results. It'll have a link to the Coriell website to get the cell line, or a link to the Adgene website to order the plasmid or get more information about that plasmid. We also have a link to our certification of analysis certificate that is also shipped with each cell line from Coriell. Now you can scroll down and you can see more information is here. So not only is that information contained in a PDF, if you want to print it out, take it to your lab bench and then take a look, or if you're on the computer, you can take a look and add it in more detail. So here we have our editing design strategy of where we tagged the GFP. We have information like the CRISPR RNA sequence, the linker. And if I click on now genomic characterization, you can see here is a schematic of the amplified junctions for our junction PCR and the results below. And just kind of walking you through here, cellular validation, we can look at, protein expression or localization by immunostaining, and then going to looking at stemness markers, we can see the state of pluripotency for those cell lines and including the differentiation potential for the different germ layers and cardiomyocyte differentiation. So we provide all our protocols also online and we update them regularly if we make any type of changes to them that might optimize the technique. And those are continuously, you can find those here through our protocols link. There's another way to get there and I'll show you in just a moment. So I'm just going to take you back to the slides. There we go. So I'm going to come back here and now I'm just going to talk about the methods and tutorials. So I showed you where the cell catalog is on our website. Now the methods and tutorials are also available. Now I alluded to the protocols or methods from here. If I come up here, I already have this saved in my browser, but you could get to it from here, going to lab methods and clicking on instructional videos, standard operating procedures or written methods or illustrated overviews. So looking at the instructional videos and tutorials. So what we've done is we've had our scientists from the different teams from gene editing and assay dev and our differentiation teams, capture some of the methods in a video format so that people can really follow the process. Because sometimes it's tricky. There's little tricks that you learn in the stem cell field about like an angle to hold your cells or, things like thawing human iPS cells, culturing and maintaining them. And we've got the RNP transfection and then we have our Jove video journal article that highlights the paper where we described our tagging approach using CRISPR-Cas9. We have a cardiomyocyte differentiation video tutorial and techniques with working with nitrogel and cryopreservation. I mean, I'm just going through and naming these. And then we also have our newly released kind of automated tissue culture platform, which if you're interested in, this kind of showcases an automated tissue culture platform to help seed and passage, feed, and maintain your cells. And so you can look for more information on here. Now, if I scroll back up and I go back to the Allen cell collection, you see also there's a link for our methods and there's illustrated overviews. So I'm going to head back. I mean, so you're welcome to look at those for more information if you're interested. And then I'm just gonna, oops. Now, our next part I want to get to before the end is our discussion forum. So I alluded to this during the talk, and you can visit our forum on allencell.org. And this forum is free to use, but we do ask that people, if they want to ask questions, to sign up for it, and which you can find here. You just sign up, you click the sign up, and it'll ask you for your email and all that, but all that information is private, stays private. But the reason why we do that is so that we don't get any like bot spam, and asking questions that aren't appropriate for that discussion. So it's just a way to keep that a safe discussion. But you don't have to log in or sign up if you just want to browse the questions and see what people have written, what topics are popular. You can hit the latest tab. That'll give you the latest topics that have been discussed, or you can pick something more specific to your interest, whether it's gene editing, culture techniques, some of our imaging software and code, which I alluded to, and I'll talk about later in future webinars. But you can just click on one of these. I'll click on gene editing, and you can see some of the top topics that have been discussed. And so that's our forum, and I think it's very useful, because what we've done with this forum is we've kind of automated any type of question that gets asked goes directly to one of our like a group of scientists or engineers to answer that question. And so they typically answer within 24 hours, pretty quick response. Now, coming back to here, I want to just kind of end with a summary of, we've created these cell lines, to answer our own scientific questions. We think we find a lot of value in this resource, and we hope you do too. And so we have been taking the step of going from large scale 3D imaging, and next we want to go towards single cell information. So here is, let me just play this here. So here's just some Z-stacks that go through some of the cell lines I discussed today. And we take images. Now we've imaged over 25 structures and counting. And we're starting to think about integrated ways of taking all those structures together. And I'll talk about some of that in the future webinars. But really what I want to kind of highlight is that, so we go from this microscopy, we take the images, and then we go through some image processing steps. So just like people that are studying single cell genomics, now we're thinking about single cell imaging. And we go through an image processing step that then can segment the cell, the nucleus, and the individual structure of the cell. And so I just want to mention that the next webinar will not be until September, but it's going to be on the Allen cell and structure segmenter. So it's going to lead into how you can take your imaging data that you get and actually start to analyze your images in a single cell way. And just think about the potential of this information, along with your single cell genomic information and other single cell information that is being widespread, how powerful this can be. As I always hear, a picture is worth 1,000 words. And so these cell lines do produce, beautiful images.